Next era of medicine. Rare disease specialist Vertex and CRISPR Therapeutics are submitting their drug exa-cel for regulatory approval in the US and Europe by the end of this year. This sets the stage for the first ever CRISPR gene editing drug to receive market authorization in 2023. Ever since the CRISPR boom, the potential was always there for it to be used as a life-saving drug. And now, it really might be happening. Vertex and CRISPR Therapeutics are applying their drug exa-cel for two different, but somewhat similar inherited blood diseases: beta thalassemia and sickle cell disease. Both have to do with hemoglobin, the protein in our red blood cells that carry oxygen. While patients with beta thalassemia have mutated or missing genes that cause them to produce less hemoglobin, patients with sickle cell disease have a mutated gene that leads to deformed hemoglobin, kinda like the shape of a sickle, hence the name. These guys can stick together, impeding proper blood flow and preventing oxygen transport throughout the body. Though the diseases aren't super common, thankfully, they are very serious and often debilitating, leaving patients in a lifetime of pain and blood transfusions. But that's where exa-cel comes in, as the CRISPR gene therapy proved to be an effective remedy for these diseases in recent clinical trial results. For 44 patients with beta thalassemia, 42 of them did not need a single blood transfusion up to 37 months after treatment. And the remaining 2 people that did require the transfusions needed significantly less blood, about 80% less. For the 31 patients with a severe and life-threatening form of sickle cell disease, up to 32 months after treatment, not one person experienced a single vaso-occlusive crisis, aka a significant stoppage of blood flow. Without exa-cel, they previously experienced almost 4 per year. These are awesome results that increase the likelihood of regulatory approval. So, how does it work? For sickle cell, it's important to know that hemoglobin is made up of 2 types of subunits, alpha and beta, and the mutation occurs on the beta subunit. This results in the red blood cells being deformed and increases the likelihood of getting stuck, which causes numerous health issues including pain. One patient has said that it's like, quote, shards of glass flowing through your veins or someone taking a hammer to your joints. You wake up in the morning with pain and you go to bed with pain. Damn. So the key to treatment is the interesting fact that when we are all infants, we all make something called fetal hemoglobin instead of the adult type. The difference is that fetal hemoglobin is made out of alpha and gamma subunits, so no beta subunits are there to potentially mess things up. And so the trick is to get sickle cell patients to produce fetal hemoglobin instead of the adult type, because fetal hemoglobin is not mutated and can do a good enough job. This is done through CRISPR gene editing of a transcription factor called BCL11A. It activates once we are born, and its job is to ramp down the gammas, like a college administration receiving one too many indecency complaints. You can see that once BCL11A is turned on, gamma is turned off, which means no more fetal hemoglobin.
If we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then, they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA-cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system, a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex. so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleases trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene, or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors, like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world.
We program the DNA to do, and it's in the living cell. Right? So in medicine, the goal in medicine now is to be able to do designer medicine and therapy. If we can design a cell to get into your body and release the right product for you, you won't be losing half the drugs you take through your liver when you swallow a pill and it gets digested. These can be inserted into you through the hypospray needles, almost like Dr. McCoy on Star Trek, giving you the hyperspray. It just blasts now plasmids into your squamous cells. But Ventnor was able to do that and has the patent on the technology. But you can engineer anything. You can engineer a unique thing that would only kill one person in the world. It's how it's done. You put in a specific gene slicing, you program what you like, you put it in the cell, and it can reproduce and make as much as you like. For those of you who don't know, your DNA is usually all wrapped up in tight little coils. And so what you're doing is when they create plasmids and put them into cells, it sends a signal and tells which portion of the DNA should unwrap, unfold, and produce a product. Now, this is the future of medicine. When you look at this technology in medicine and say, this is going to be done to help people, right? We want to be able to give them medicines. We actually want to correct for genetic deficits. If a kid's born with a genetic anomaly, with the CRISPR technology, the feeling is we can create the portion of the gene they're missing and go have it spliced back in. And that may help a child, either if it's in utero development or once they're older, to have the missing substance actively produced. What would you do with this if you were in security and intelligence? Well, you can do a number of things. You could decide, if you make this gene, we know that certain people in the world who function at very high altitudes very, very well do it because they had a special mutation in their genome that we don't have because we didn't grow up in the Himalayas. But they can function at very high altitudes. Could you give this to people who are going to have to do warfighting in high altitudes and they don't require extra support? Their body makes a much more efficient use and can work under conditions of lower oxygen than the rest of us. You start letting your mind wander. Can it also produce a substance that lets you function longer underwater without oxygen? But these are run by certain mutations in genes, and with CRISPR we have the ability to actually make these and see what happens when we give them to animals, non-human or human animals, that don't have it naturally. You could have the Forrest Gump gene that just makes you stronger. I would say that most of this technology is probably going to be employed by a state and not non-state actors because it's quite technical. But I say that with a caveat. When we studied Shinrikyo, if people remember, they had both uranium mines and regular laboratories where they experimented on both animals and had a whole series of laboratory experiments to develop the different kinds of gases that they wanted. Their goal was to actually mine uranium and probably come up with their own version of a nuclear weapon. But they recruited scientists, PhD level folks, and their goal was to be the rightful people running the country of Japan. But we can't assume that just because they're non-state actors, they will not make use of some technology around this. Related to this is an idea called DREADDS. These are designer receptors.
It's going to transcribe and translate proteins, so this Cas complex, and it's also going to transcribe that DNA to make what's called CRISPR RNA, and it'll fit right into this protein like this. What is this? It's a way to fight that viral DNA. It essentially breaks it apart, and so before the infection starts, the infection essentially has ended. Now you might say that's interesting, but what happens if it's injecting DNA where we don't have a spacer that matches? Well, the CRISPR-Cas system works there as well. It's going to create a different class of protein, a class 1 Cas protein, and what that'll do is it takes the DNA in, it breaks it apart, but more importantly, it takes that DNA and copies it into the CRISPR system. So what is CRISPR? It is spacer, repeat, but the spacers are essentially history of old infections so we won't be infected again. This is exactly the way your immune system works on a much larger level. You're making antibodies and then you have white blood cells that'll envelop that invader. But what scientists thought is if we could hijack this CRISPR system, we could perhaps use it, because this is a living cell here, to either inactivate genes or maybe even embed new genes. And so the search was on. And the one that you'll hear most about is the CRISPR-Cas9 system. This was identified in the labs of Jennifer Doudna and Emmanuel Charpentier. And what she was working on was Streptococcus pyogenes and their Cas CRISPR system. And what's interesting about it is that they only had one Cas protein. We call that Cas9. Now it doesn't look like this, it looks like this. But if we look at its major structure, it has a nuclease. So it's got this section right here where it can cut DNA here and it can cut DNA here as well. In S. pyogenes, they also are creating two long strips of RNA. We have the CRISPR RNA. The CRISPR RNA is going to fit into the Cas. But they also have what's called tracer RNA. So if we look at what that looks like in this bacteria, you've got the spacer segment. That's going to be the part that matches up with the corresponding viral DNA. You have this tracer RNA that essentially holds the CRISPR RNA in place. And then this whole thing together forms this complex where we can break DNA. But what the lab thought is, wouldn't it be cool if we could modify this whole system? Use the one Cas9 protein, but let's put our own sequence of DNA right here. And then if we could somehow connect these two together, we'd have a really simple system. And that's what they did. They created the tracer RNA, CRISPR RNA, chimera. And so what's a chimera? It's this ancient mythological beast that's a combination of all these different species. And so what they've done is created a new type of RNA. And they've got a system that's really simple. It's got two parts in it. You've got the Cas9 protein, and then you've got this chimera. And since we're making this simpler, let's just call this the guide RNA. These are the two parts of a CRISPR-Cas9 system. This is going to be the CRISPR part. It's going to be the RNA that's got the information of where we want to cut. And then we've got the protein that's actually going to do the cutting. And this is what happens. And so if we've got a little bit of DNA, so this is the DNA that we want to cut, we create a guide RNA that's going to have a corresponding bit of RNA. what happens is the DNA will feed into it like that. Once it's in place, we're going to cut it right here, and we're going to cut it right here. And so we do this little snip, and now we have an inactivated gene. We've broken the gene. Now the cell will try to fix it. It'll do some insertions and deletions, creates mutations. But what we can do a lot of the time is we can inactivate that gene. That's what the bacteria are going to do. But since we've created it, we can cut the DNA wherever we want to cut the DNA. We essentially just have to know what is the sequence of DNA that we want to cut, put that into the guide RNA, and then we can cut it. Now let's say we want to make this more complex. Not only do we want to break a gene, but let's say we want to insert a new gene. Well, now the system's going to just have three parts. We've got the Cas9, we've got the guide RNA, and then we've got the host RNA that we want to put in. So as we break the DNA, the host DNA is going to be added, and then the DNA is going to fix it. So essentially we've added the gene to the cell. Now what's cool about the CRISPR-Cas9 system is it does this in living cells, and it can cut the DNA in multiple different places. So how could we use this? Well, let's say somebody has cystic fibrosis. What we could do is use a system like this to fix the genes in that person.
There's one more question that a lot of people have about CRISPR-Cas9. I remember when I was learning about CRISPR-Cas9, this really bugged me, right? I understood why Cas9 doesn't cut the bacteria's own DNA because it lacks the PAM site, and it needs that. But why doesn't Cas1, Cas2, why don't they cut the bacteria's own DNA? Why don't they collect a protospacer out of the bacteria? Because all they need is the sequence, any nucleotide GG. Surely there's lots of those in the bacteria's own chromosome. Surely. So why don't Cas1 and Cas2 find two guanines together and then cut a protospacer out of that and turn it into a spacer in their CRISPR array? Well, it turns out that the reason is because of those chi sites. Remember, E. coli, and other species of bacteria too, no doubt, are full, their chromosome is full of chi sites. There are chi sites everywhere. Now, of course, the reason for those chi sites, the main reason for those chi sites is that so that enzyme REC-BCD that resections the 5' end for double-stranded break repair, that's what those chi sites are there, but they serve another function too. Because as it turns out, Cas1 and Cas2 won't make a spacer out of DNA that contains chi sites. And because there are so many chi sites in a bacteria's own DNA, that means that Cas1 and Cas2 pretty much don't make spacers out of the bacteria's own DNA. Whereas bacteriophage DNA has little to no chi sites in it at all, which means that Cas1 and Cas2 will make spacers out of that, but not the bacteria's own DNA. And this is another wonderful example of how this adaptive immune system of a bacteria is able to distinguish self from non-self.
Walter Isaacson's new book, The Codebreaker, tells an amazing story. It's about how Jennifer Doudna and other scientists discovered a new way to edit DNA. Editing DNA with precision has been a holy grail for scientists for decades. The DNA, of course, encodes all of the biological functions. And so some of the mistakes in DNA cause disease, genetic disease. If we could go in and fix those mistakes, we could save many, many lives and get rid of these diseases. CRISPR is a very fascinating tool. You figure out a part of the DNA sequence that you want to change. Then you take the DNA that's nearby and create a guide in RNA, so that helps you locate the position. And then you link this Cas enzyme, which has the ability to do that cut and edit. And so pulling together this guiding RNA and this Cas enzyme, editing becomes possible. Our foundation is funding work to see if we can use CRISPR to knock down mosquito populations dramatically. We're also looking at CRISPR to make better seeds. We're also looking at ways that CRISPR might help us with very accurate diagnostics. And we're considering how the CRISPR gene editing might lead to a way of curing HIV and things like sickle cell. Using CRISPR to help cure disease is not very controversial, but using CRISPR, you could actually change the DNA that would determine your baby's eye color or skin tone. Most scientists agree that this is something that we should not do. Walter's book does a great job talking about how it's a slippery slope and raising the question of where should we draw the line and the relative role of the scientific community and governments in helping make sure that we don't cross it. There's no question CRISPR is going to save a lot of lives. It's going to raise issues.
To help explain RNA editing, let's think back to how books were printed years ago. First, a manuscript would be created. The manuscript would then be sent to a typesetter who would painstakingly put the type in place. Then, an editor would proofread the typesetting and fix any errors. The book could then be printed. Now, imagine the original manuscript is the MECP2 gene, the typeset is the RNA, and the printed book is the MECP2 protein. Now, what happens if the MECP2 gene has some mutations? We can think of them as typos. All of us have an enzyme in each and every one of our cells called ADAR that acts like the editor, proofreading and correcting mistakes in RNA. We can actually hijack this naturally occurring process and put it to work to fix typos in MECP2. The lab of Gail Mandel at Oregon Health and Science University in Portland has already shown that they can introduce specific RNA guides into a petri dish of ret mouse neurons. These guides escort the ADAR to the typo, where it can get to work correcting it. Cool, right? The Mandel lab is now repeating this experiment directly in mice to see whether their symptoms can be reversed. RNA editing is specific to a particular MECP2 mutation, so a library of these RNA editing therapeutics would need to be developed. One advantage of RNA editing is that you never end up with too much MECP2, which could be a problem. RNA editing is a major focus at RSRT, and we're also funding other labs that are using different approaches to achieve the same end result. The more shots on goal, the better. These experiments are made possible through RSRT funding. Your donations hard at work.
Bright as well. According to a drug development program within MIT, though it's not all CRISPR, they anticipate over 60 gene and cell therapies to be on the US market by 2030. I know we hear a lot of bleak stuff about the future,